Hello, everybody. Hopefully, you had a nice few days break. I thought this week we could do something a little bit different and try to do some sort of at-home kind of experiment using microorganisms. Now, I call it microorganisms because instead of just bacteria, hopefully it would be bacteria. But when I did it, I got mainly mold, which is a fungus, and who knows? It could be fungus, could be mold. We don't really have the ways to test it at this point. So it's just me microorganisms, and I thought it'd be kind of fun to do a little experiment and see what we come up with. Be creative. So first off, you should have your parents' permission. Make sure they know what you're doing and that it is from me. And don't make a mess, please, or get anybody sick, or die. No, that would be bad. So let's keep this safe and let's try to be fun. So I'm going to go through what I want from you and how to do this. And if you have any questions or anything, just please let me know. The sooner the better, because this will take a few days to get to really get through. I think the time-intensive part will be the beginning and the end, and then the middle. You're just taking pictures, so it'll be all right. All right. So our goal is to compare microorganism growth under different conditions. And we're going to start, you want to brainstorm about what you want to do as far as your hypothesis. And, oh, sorry, I don't even know my slides. All right, so what we're going to use to do this, I had to take a poll. I think like 95% of you have Jell-O or some sort of form of Jell-O at home or could get it. So I thought about this later, and I didn't use these, but these Jell-O cups already kind of have this built-in lid that you can use. So if you have that, awesome. If not, find Jell-O or gelatin. And if you don't, there are other things you can use, such as Crisco, which is almost like a lard, just like a grease, bacon grease. We have a grease jar at home. You can try that. Get some grease out of that. Something with a solid surface and with nutrients for microorganisms to grow. Typically, they use a substance called agar, which is already pre-mixed with nutrients and everything that it needs to grow, and you can put it on a plate and it's all nice and neat, but we don't have it at this point. So something with a somewhat solid surface. I would even say like you could maybe, if you're in a pinch, use a pudding, although it's not quite solid. It'd be hard to do. You're going to want 3 containers with lids. I used a kind of a Rubbermaid Tupperware container and an old sour cream container with a lid. You got any kind of pasta sauce jar, jelly, peanut butter jar, any of these jars. One thing you want to try to do though is make sure is that you use something that's been cleaned out. So you want to try to make sure that there's no contaminants as much as you can. That just occurred to me that you could use peanut butter as well. It's a pretty solid surface. And then you're going to want something to compare it with. What I ended up doing was I did Jell-O, and then I put some beef broth or beef bouillon cubes into another Jell-O, and I compared growth of both. You can take the Clorox wipes and you can cut them into little pieces and put, you have one regular that's inoculated and one that has been inoculated with the wipes, and see if there's a difference in growth around the wipes. You can take a Clorox spray and spray it. You can go outside and get, put a Q-tip in some dirt, and then put a different Q-tip in your mouth and see if there's a difference in growth between your mouth and the soil. I mean, really, your comparison and the kind of the third material here is up to you. That's where your creativity is going to come into play. All right, so first you want to figure out what you're going to test. And there are so many different options here. And today, what I want you to do is kind of brainstorm and figure out what you want to test, and then fill out the question on Jupyter and the Junipod so that I know and I can see and kind of clear you for that to make sure that it sounds good. But there's many different things you can do. Like I said, Jell-O versus Jell-O wipe with Clorox. You can do sugar-free Jell-O versus regular Jell-O. Is there a difference in growth there? Location, toilet versus sink. Soil versus carpet. Dog or pet saliva versus human saliva. Cold versus cold in the fridge versus warmth. If you have some antibiotics laying around, you can crush those up and throw it on there. Again, get your parents' permission for that. Anything really, and I would hope I'm looking for something that's kind of creative, that's kind of thinking things out a little bit. Um, Something to, I don't know, something you're interested in. But there are plenty of things you can do. These are some ideas. You are not limited to these ideas, but you're going to want to test two things, and then you're going to have a control. So once you figure out kind of what you want to do, what you're going to test, then think about, well, what will those conditions be? All right, determine the question you're going to be testing. One question would be Clorox wipes will inhibit microorganism growth. All right, that's a cool hypothesis. Hopefully not 95% of you take that one, but, my hypothesis for the one I did was that beef bouillon, adding that would allow the microorganisms to grow more quickly or grow better than without. And I will say that the jello that I put beef bouillon in and mixed that all up smelled pretty funky. It was not a pleasant smell, but it did grow. And once you get your hypothesis, you're going to want to record that. That's because we're going to write a final report at the end, so you want to keep all this documented, and it shouldn't be too bad. Step 3, you want to get your materials, your jello, your 3 containers, your libs. Maybe you need Q-tips. What I did was I took the jello, and then I put a Q-tip, the cotton part in the mouth, and got saliva. There's plenty of bacteria in your mouth, and use that. But there's also fungus growing in the air, so you can do that as well. I mean, I don't know. But you're going to need 3 containers because one of them will be your control. You're going to put whatever your material for growth is in it, and then just leave it alone, and except outside of taking pictures. This is the control is the one that you have that is where it's not changed, it's unchanged. So your material is pretty basic. Again, depending upon, you could have other things in there as well, whatever that difference is between them. All right, then you're going to make step 4. You're going to perform the experiment. Hopefully you get that done. Maybe tomorrow you're kind of brainstorming, and then Thursday you get it all made. And so you're going to make your jello, and then you're going to inoculate with your microorganism source. I did this a little bit different. So I used saliva, and you can just going to put a loogie on there and spread around with a Q-tip, or you can put the Q-tip on your mouth. I don't know. It sounds kind of gross, but we want microorganisms, and we want them to grow. And hopefully there's something at the end we can kind of state whether something happened or not. You're going to want to label the container so you don't forget, and put the lids on. It's important because there are microorganisms all over the place. You want this to be as controlled environment as possible, and they're going to store it. I had mine in the basement, which was a little colder, and it, I think, inhibited the growth a little bit. But store it in a place that your parents think it's cool. You don't want it on the dinner table. And then you're going to document it. So you let it set, and I will say the jello does take a few hours to set. And you want that to kind of set before you do anything. So keep that in mind. And then you're going to document it. You're going to take photos, I would say at least once a day. I took them once, sometimes twice a day. Hopefully at the same time. So same time of day and at the same time, all 3 of them. Do keep in mind that you want to do that as quick as possible, as to not contaminate it with some sort of fungus that's growing around. But it'll happen. But take photos. Mine took about 6 days before it showed any growth. I'm hoping that if you have it at more of a warmer temperature, it'll work more quickly, or better. Maybe I just have a clean mouth or something. I don't know. And then you're going to write the lab report. Tentatively, it would be due Monday. It'd be your work for Monday. And then we'll start something new on Tuesday. Then there are 5 sections: your hypothesis, your materials, your procedure, your data, your conclusion. And I have a separate document outlining that and how you do that. It's pretty straightforward. I mean, really, the first 4 kind of do them. I mean, they kind of write themselves. Basically, just uploading pictures onto the paper, and then the conclusion is just writing up what happened, right? And the conclusion a little bit more. You're going to have a little bit of leeway in there as far as the creativity of the experiment. How well you explain. I don't want you just to point and say, hey, picture 5 shows there's growth, and picture 6 shows no growth. State your hypothesis, explain whether or not you think that was correct or not, and then explain why. why you thought it was correct or did not think it was correct, the reason behind that. And again, I have a sample kind of report that I pulled up explaining it and giving you my from my experiment. And I can show you now. I have pictures. This file. So these are my files. You can see that I had a little bit, day 1, no growth. I will let's see if it say that, and that's day 1 for the beef brewing. Again, just nothing. Day 1. Day 2, you can see the little dots in here. I suspect those are bacterial colonies, but they never really got very colorful. So I think I had a pretty good bacterial growth. But what I ended up with is fungus. This is day 2 with the beef broth. And as I'm doing this, and I was unsure of whether or not I wanted to go with this, so I kind of put it on the shelf for a little while. But day 5 for the beef broth, I think that's the glow from the camera. I don't think there's anything there. And then day 6, there started to be some growth, and I think it's fungus just by the fuzzy and furriness of it. Probably something that came from my basement, but it looks good. And then this is day 7 with no broth, and the colonies are kind of beginning to show there, but I ended up cutting it off, I think, at day 7. Day 7 looks really good. And then day 8, you can see there's some colonies growing there. And then I think that's day 10. I kept going to that one. It was a slower growth. But as you see in my lab report, I kind of fudged it a little bit. I just cut it off at day 7 and went with that. Hopefully it doesn't take as long for you guys. Hopefully just 4 or 5 days will be good. Again, shoot me any questions you have. It should be kind of fun. Hopefully it's more entertaining than listening to me talk every day. So that is that, and good luck.